Okay, so welcome back to this next video in which we are discussing the reduced SERCA function in pathological cardiac hypertrophy. Okay, right. So we have made this prediction that if we return the SERCA2 pumps into the membrane of the SR lumen, into the membrane of the SR, then we should see that the hypertrophied cardiomyocyte, that its ability to generate force is returned basically to normal. Okay, so let's have a go. So we're going to do this by adenoviral gene therapy. So let me just explain this process to you. So adenoviruses are a type of virus which has a double-stranded DNA genome. So let me show you the structure of an adenovirus then. Adenoviral gene therapy is what we're discussing. So we're going to use this to implant in an extra gene for SERCA2 into our cardiomyocyte so that the production of the SERCA2 protein will go up. Okay, so the structure of an adenovirus then. So it has a capsid around the outside which we'll draw as a hexagon, but in fact it's some platonic solid. It's usually an icosahedron or a dodecahedron, but we'll draw it as a hexagon since we can only draw in 2D. Okay, so this is the capsid and it's made up of proteins. So this is the outer shell, if you like, of the virus. So this is the capsid. Then on the outside of the capsids you have these little proteins which are sort of pointing off the sides. Okay, like so. So you have these, I'll draw them at every single corner is the way it's usually done. Okay, so in the real virus what you'll have is more corners than 6, and at all of the corners you have these little proteins like so. Right, so this portion that attaches the extension, this process to the capsid, is what's known as the penton base. Okay, the portion in the middle is then called the fiber. Okay, so this is the fiber, and then finally the portion at the end is known as the knob domain. And this is important because this is how these processes which stick off from the capsid of the virus are how the virus is going to stick to our cell. It's going to bind to receptors on the cell membrane. Right, okay, now adenoviruses do not have a viral envelope. Other viruses, such as for instance HIV or influenza virus, they have a viral envelope, a membrane around the outside of the capsid. In the case of adenoviruses, they do not have a membrane around their capsid. Inside the capsid, they then have their DNA genome. So for adenoviruses, it's a double-stranded piece of DNA. So this is often denoted as dsDNA. So the little ds means double-stranded DNA, and then genome. Right, so this piece of double-stranded DNA has all their genetic information for making viral pieces in it. So I'll colour in this DNA in orange here. Okay, and I might just colour in the capsid as well. So I'll colour in the capsid in this green colour here, or turquoise colour actually. And then maybe I'll colour in these processes also. So in blue then we have these penton bases which are attaching to the capsid, followed by the fibre, followed by the knob domain right at the end. Okay, and you have these at every single corner. Now if you're dealing with a platonic solid, you'll have far more corners than I've been able to show you on this piece of paper. So in reality this is a 3D structure and it will have loads of corners and it will have one of these at every single corner. Right, okay, so now let's discuss what we're going to do. Basically we are going to take one of these adenoviruses here, so this is an adenovirus, and we are going to remove that adenoviral genome, okay? So we're going to take its DNA out, chuck that away, and put in a new piece of DNA which has the gene for SERCA2. Okay, brilliant. Now what we're going to do is unleash this virus on our concentric hypertrophied cardiomyocyte. So what will happen is, if we draw our cell here, the adenovirus will come along and it will bind to receptors on the cell. So where should I draw this? I'll draw this here maybe. So here's a receptor on the cell, and now here comes our adenovirus, which I'll just draw as a little hexagon now, and one of these little processes, which I'll just now draw as lines coming off, has bound to the receptor here. Okay? Now, when it binds to the receptor, when this process in blue, so I'll draw it in blue so that we know what we're talking about completely. So these are all these blue processes consisting of these penton bases, these fibers, and then on the end the knob domains. And finally, just to complete the picture, I'll colour in this hexagon in turquoise for the capsid. Right, okay, so the adenovirus has come along to our cardiomyocyte. It's bound to a receptor on the surface of the cardiomyocyte membrane. Then what will happen is an adapter protein, which I'll just draw as this, will come and bind here, so some adapter protein, and specifically it's adapter protein complex too, so it's not just one protein, in fact it's a complex of proteins. So AP2 is short for adapter protein complex 2, and this is the structure which will bind to the receptor once the receptor has bound to its ligand, and this is what's going to result in this receptor and the viral particle attached to the receptor being endocytosed. So then what's going to happen is clathrin is going to come and associate with this adapter protein complex 2, and then it's going to polymerize. Okay, and if I draw this process up here, so we'll show another one further along the process. So what's going to happen is, here is our receptor, here, with the adenovirus bound to it here. So I'm just showing receptor-mediated endocytosis here. So you have your adenovirus still bound to the receptor in the middle here. Okay, so let's colour everything in so we know what we're talking about. So here's the receptor in red. Here's our adenovirus here, and here are these little processes in blue, and the capsid in turquoise. Okay, and then some adapter protein complex, this adapter protein complex 2, has come and bound here. Okay, so I'll show that in turquoise, sorry, not in turquoise, in vivid purple here. Okay, and now what's happening is the clathrin protein is polymerizing. So initially one clathrin protein bound to the adapter protein complex, but they're all joining on, and when they joined on, when they polymerize together, they form a spherical-like structure, and so they pull the membrane in around them as they form, and this is creating this clathrin-coated vesicle bud, as it's called. So this is a clathrin-coated vesicle bud. Okay, and then the final portion of this process is just to pinch off this clathrin-coated vesicle bud, to pinch this membrane off here, to make it thinner, and then separate this vesicle off from the plasma membrane, and that process is undertaken by dynamin proteins, and also endophilin proteins, so they're responsible for the budding off process. Then what you have, they're not responsible for the budding off, they're responsible for the pinching off. Clathrin is responsible for the budding off process. Okay, so then what you have is a clathrin-coated vesicle within the cytoplasm of the cell. The clathrin will then be removed, and what will happen is the adenoviral particle will break out of its endocytic vesicle. So it will break out of the endocytic vesicle, and what it will then do is it will go to the nucleus of the cell. So let's say here is the nucleus of the cell, and it will implant this DNA into the nucleus of the cell. So here's our adenovirus here. And now it's at the nucleus, and then it will put the DNA inside the nucleus. So here comes the SERCA2 gene that we had implanted inside the adenovirus, and now it will be inside the nucleus. Now, an important point is that it will not insert it into the actual genomic DNA of the cell. it's just going to stick this extra piece of DNA into the nucleus. So all the adenovirus does is it delivers the DNA to the nucleus. It does not integrate it into the DNA of the cell. So it doesn't chop up the DNA of the cell and then stick in this new piece like a retrovirus would. It just sticks it in alongside all the other pieces. Now the idea then is that the cell will use this piece of DNA like the other chromosomes, and it will make the SERCA2 pump from this piece of DNA. That means that the amount of SERCA2 pump that this cell is making will go up. So you're going to have more SERCA2 pumps in your sarcoplasmic reticulum. And what you find is that this is actually extremely successful. If you do this to concentrically hypertrophied cardiomyocytes, you see that the size of the calcium spikes upon the cardiomyocyte receiving an action potential are bigger. So the calcium spikes go back up, and this is because the level of calcium within the SR has gone up as well. So the level of calcium in the SR goes up. Calcium in SR fundamentally goes up because you've put more SERCA2 in the membrane of the SR. That causes the calcium that's being returned into the SR to go up. So calcium in the SR goes up. That means that when the cell undergoes an action potential, i.e. when when the heart's beating, the sinoatrial node fires action potentials along all of the cells, and it's going to arrive at our cardiomyocyte. So when an action potential arrives, that will cause the calcium spike, and the calcium spike is going to now be bigger. That's then going to mean that the number of sarcomeres that we recruit is going to go up. So the number of sarcomeres that are actually recruited, that are actually going to contract in response to this calcium spike will go up. So if we've got more sarcomeres contracting, then the force that we generate is going to be greater. So in fact, returning the SERCA2 pump into the concentric hypertrophied cells is extremely effective at returning the force that these hypertrophied cardiomyocytes are capable of generating back up to normal. Okay, right, so that's a nice experiment showing proof of principle then. What we'll now turn our attention to is the other part of downregulation of SERCA2 function. So one thing that leads to too little SERCA2 function is that you just have too little of it in the SR membrane. And we've just shown that if you increase it, that seems to be an effective treatment. And there are problems with actually doing gene therapy in vivo, but we won't go on to those. There is, however, another problem with SERCA2 function, and that is that it's being inhibited by phospholamban. So we'll discuss phospholamban in the next video.